Hi, my name is Meredith Ashby, and I'm the Director of Market Strategy for Microbial Genomics here at PacBio. And I'm going to talk to you today about the HiFi sequencing advantage for metagenomics. If you'll just give me a moment to start up my slides, we can get started. Okay. So today I'd like to start off with a review of HiFi metagenomics and then talk to you about two specific use cases to really dive into how HiFi can make a difference in your metagenomics research. The first is a use case involving 16S sequencing, looking at pathogen tracking in NICU infants. And the second is sequencing using HiFi and looking at fecal microbiome samples in the context of a clinical trial for the evaluation of live biotherapeutic. So what is HiFi? HiFi are unique to PacBio, and they combine long read lengths with very high accuracy. The way we do this is by generating multiple passes across a single template molecule, and then combining those observations in order to generate one consensus read that has an accuracy of over 99%. So that's accuracy that puts it on a par with what you typically would get with short read sequencing. So there are three different ways you can use HiFi sequencing in order to study metagenomics with the PacBio platforms. So the first is full-length 16S or even longer amplicons, for example, with the strain ID kit from Shoreline Biome. So these longer amplicons allow you to distinguish species and strains from genus-level noise. And that deeper information will allow you to generate better and more testable hypotheses. On the Sequel II system, you can get about 30,000 reads per sample at 96 plex. And if you use the simple all-in-one kit from our partner, Shoreline Biome, you can have a complete end-to-end solution that includes DNA extraction, PCR amplification, sequencing on one of the PacBio platforms, and then analysis using their custom software and database that's specifically annotated to do well with long read lengths. A second application is metagenome profiling. So HiFi sequencing is a very powerful tool for metagenome profiling because, as you can see from this table, the average read length that you get from HiFi is between 8 to 10 kb in length. And that means that there are multiple genes, sometimes as many as 9 or 10, complete, intact, and error-free genes on every read. And because of that high accuracy, they're very easy to annotate. So unlike with short read solutions, 80 to 90% of HiFi reads will have at least one functional annotation, with most having 2 to 4. And another strength of HiFi is that since you don't need to do error correction, you can see metabolic functions even from species that don't have enough coverage in your data to do assembly or to do error correction. A last thing that HiFi reads are really great at is mag assembly. So everyone knows that the first step in a metagenome analysis is typically to do an assembly, and it's pretty clear that long reads in general improve assembly results, but not all long reads options are the same. If you have noisy long reads, these first have to go into an error correction step before they can be used for assembly. And the challenge here is that in metagenomes, it's typical that you will have many strains and species that are very closely related. And in fact, the difference between any one of those two closely related strains or species is often less than the error rate of those noisy reads. So what happens when you go into error correction is that those real differences that you need in order to correctly assemble nearby strains is lost and averaged out because those true differences can't be distinguished from the error rate, which is actually higher. Unlike noisy long reads, HiFi reads are very accurate, so you don't need to do this error correction step. And all of those unique differences between closely related strains are preserved. And that allows you to get better strain resolution from your complex metagenome sample. And it improves the assembly completeness because you don't end up with assembly graph tangles that cannot be resolved. And then finally, you have much higher contig purity. If this sounds interesting or exciting to you, you should check out our recent blog from October 21st. In this blog, we have presented some modeling data that was done by Richard Hall, a bioinformatician here at PacBio, who's looking at how this idea of noisy versus highly accurate long reads plays out in terms of assembly efficiency when looking at complex microbiome samples. So with that, I'd like to move into our first use case, tracking closely bacteria at high resolution in NICU infants. So the first point I'd like to make here is that the strain is really the functional unit of the microbiome. If you take this very simple use case, this E. coli here does not produce any toxins and is a commensal, and you're at no danger if you have this E. coli in your digestive tract. On the other hand, this E. coli is a bad actor and is multi-drug resistant and has made many people sick. But both of these E. coli will just come up as Escherichia shigella using common partial 16S sequencing assays. The good news is longer amplicons with more than 16S information can adequately resolve these and very well. And that's great news because many studies have shown in many diverse areas of research, including cancer, predisposition, and autoimmunity, that the strain is really important because two different strains from the same species can in fact have quite different impacts. So what is the right length of amplicon to really be able to differentiate strains one from the other? So if you look here at this E. coli species, you'll see that this E. coli, like many bacteria, has multiple copies of the 16S gene, right? And so one thing you can do is to leverage this diversity in order to generate a unique assemblage of ASVs that together constitute a footprint, which will uniquely distinguish this E. coli from other closely related strains. And what should the length of that amplicon be? So if you sequence only the V4 region, you can see that every one of these different copies is completely identical. So that's not going to be enough. And if you look at the 16S region, there is some diversity that's captured here. But if you look a little further out to this ITS domain, which separates the 16S from the 23S genes, this is really the mother lode of diversity. And so if you sequence an amplicon, the entirety of the 16S gene includes the ITS domain and then is anchored in the 23S, again, more conserved region, you can really do a great job of coming up with a unique assemblage of amplicons for every genome that can be used to fingerprint the bacterium. And that's exactly what the strain ID kit from Shoreline Biome does. It generates multiple unique amplicons per genome that can be then used to fingerprint strains as they appear in the microbiome. So here's a practical example of how that plays out. So Shoreline Biome worked with some collaborators at the University of Connecticut to look at strain tracking in NICU infants over time. So this is interesting because first of all, premature infants are born with a compromised intestinal barrier in their gut. And what can happen is that bacteria can move from the gut into the bloodstream and result in very serious illness. So one thing people want to know, of course, is if there's a child in the NICU, are there pathogens that are coming in? And where might those be coming from? Are they being transferred from other infants in the NICU? Are they coming from the hospital environment? And this is a kind of detail that you really do need that strain level information to answer. Another thing that people are very interested in studying, of course, is the development of the infant microbiome over time. So the first input to the infant gut microbiome is probably the birth method. But then, of course, the hospital environment can contribute, and then nutrition sources, whether that's breast milk or formula. And another impact that can happen in the NICU in particular is antibiotics. And we all know that antibiotics, when they're given, are life-saving, but they also can compromise the gut microbiome. Right? And so they wanted to look at how these infants are faring over time and how that complex microbiome community is developing while the infants are in the hospital. So for this study, they looked at the microbiomes of two twin pairs, with each twin pair being hospitalized in the same NICU. And with the two pairs existing in different hospitals. And in addition to that, they included in the study other infants that had been in each hospital at the same time. So they obtained fecal samples for 8 to 9 weeks in order to do a longitudinal assessment of the population post-admission. And what was a surprise was that all of the samples actually contained suspected pathogenic strains of well-known species. So if you look here at the data for E. coli strains, what you can see is that there are at least 3 different fingerprints, unique assemblages of ASVs that appeared in these infants, right? So pattern 1 is shared between twins A and B in hospital 1, and then twins Y and Z in a separate hospital each had unique and different E. coli strains present, right? And the great thing is that these fingerprints are quite distinct and easily resolved one from the other. So even though these are novel strains and they're not named and we may not have the entire genome for these, using just this distinct ASV fingerprint, you're able to track the presence of these over time in these infants. And in this case, you can tell that there was likely no transmission amongst these infants over time, with the possible exception of twin A and B, which may have happened very early in life or possibly during birth. Here's a second example, now looking at Klebsiella. So Klebsiella is, of course, another dangerous pathogen that is commonly spread in hospitals. And this, once again, the strain ID Amplicon was able to detect unique ASV fingerprints that can identify and track novel strains. So in this case, you'll see that twins A and B both shared the same Klebsiella strains, the same ASV fingerprint, whereas twins Y and Z also shared a common strain, but a distinct one from the other twin pair. And then you can compare, looking at this now larger data set with more infants, that infant PC over here and PX had distinct and completely unrelated strains as well. And that all of these strains differed from known strains, which are shown here in order to show the contrast. And again, the point here is that each of these different strains was readily identifiable and distinguishable one from the other because these amplicons do include that highly variable ITS domain. And so this is a way that looking forward will be a very powerful method to really start to disentangle how one strain impacts the gut microbiome while another one impacts it perhaps in a different way. So moving on to our second use case, this is a story about HiFi sequencing of fecal microbiome samples in the context of the clinical evaluation of live biotherapeutics. And I want to acknowledge that the work that I'm showing here was done by my colleagues Shreya Chakraborty and Dan Portik, and also Richard Hall. So for this project, we worked with an industry partner who came to us wanting to collaborate and explore whether HiFi sequencing could help them do a better job of getting the most information that they needed out of these samples that they had collected during a clinical trial. So this industry partner is interested in developing live biotherapeutics to address chronic inflammatory disease. Right, so chronic inflammatory disease is, of course, a huge concern, both allergic disease, the increasing prevalence and severity of allergies in young children, metabolic disease, which leads to type 2 diabetes. And then in addition, another area that they're interested in looking at is post-antibiotic recovery. So they told us after their first clinical trial that they had collected short-read data, but they wanted, again, to see if they could answer more questions using HiFi. So in this case, what they really wanted to do, their key needs, was to be able to track the engraftment of 3 different probiotic strains that are delivered together as part of this biotherapeutic. And they wanted to also be able to distinguish those strains from closely related strains that may have already been in the gut of the patients, or which may arise in the gut or grow out in the gut upon treatment with this probiotic. And then finally, they wanted to be able to assess how the functional profile of the gut changes upon treatment. And so this means not just looking at the function of the biotherapeutics they delivered, but also how all of the other species in the gut may have changed and evolved, and what new functions have appeared or disappeared as a result of the treatment, and how those might be connected to future changes in disposition to allergic disease, metabolic disease, and other syndromes. So the way we designed this pilot is that they chose 5 different patients and they had provided us with pre and post-treatment samples from those 5 patients. And again, these samples were collected in the previously done clinical trial, so this was all retrospective. And we multiplexed 4 samples at a time on a single SMRT Cell 8M, generating a mean of 5.9 gigabase of data for each sample. And this is what the HiFi data looked like at a high level. So you can see here all the different patients, and then you can see that the median read length for almost all these patients was between 6 and 7 kb. I do want to call out there is one patient, this is the patient 3 pre-treatment, who did have insert sizes that were a bit shorter than average, because that's going to come up in another slide. And then you can see finally that for most of these, we're getting about 1 million reads per sample, which may not sound like a lot if you are used to the number of reads that you would get from an NGS experiment, but remember, these reads are very long. So for the analysis, we did assembly with HiCanoe, and then genome binning using Minimap, MetaBat2, and CheckM. And then finally, to do taxonomic and functional annotation, we used Diamond and Megan 6. So more detailed information on all of these pipelines can be found at the PacBio GitHub page. So here's the high-level assembly results from Canoe. And what you can see straight away is that for all of these different patients, we're getting some very high-quality assembles. So there are on the order of 20 to 40 contigs that are each greater than 1 megabase. And there are a number of circular contigs within the assembly as well. And those circular contigs can either be chromosomes or plasmids. And then finally, if you look at this last column, you'll see that the longest contig for most of these is very large and could easily be a complete strain. So let's dig in a little more to some of these high quality mags. So on this chart, we have in blue all the high quality mags, by which we mean greater than 70% complete, less than 10% contamination, and fewer than 10 contigs. And then in addition, we have in green all of the single contig mags. So what you can see right away is that the HiFi read link really allows for more contiguous assemblies, because we're able to assemble through repetitive regions, and not just repetitive regions in a single bacteria, but all of the repetitive regions that are shared and common throughout the entire metagenome sample. And then secondly, you can see, and what you can see is that this patient sample that I flagged before is having shorter read lengths than average, patient 3 pre-treatment sample. You can see that if the reads are not long enough, you do in fact have some compromise in your ability to generate mags. So the length of those reads is quite important. Second thing that's super important here is the single molecule accuracy, right? So with this high accuracy, that means that we can distinguish reads that are coming from closely related species. And if you recall, typically the definition of a different species in bacteria means a 97% identity. That's the cutoff. And so because HiFi reads are 99% accurate and higher, that means these reads are not averaged out. The true information that you need to get those better mags, to get those better assemblies, is not erased during any error correction step, because there is no error correction step. So digging in a little bit more and looking at just one of these patient samples in terms of the assembly results, you see on the left-hand side the unfiltered bins from MetaBat2. And you can see that in general, there's very low contamination for all these HiFi reads. And then if you subselect out just the high quality mags, with the cutoffs that I described before, you can see in this sample there are 27 distinct high quality mags, and 18 of them are greater than 98% complete. And 6 of those are present in a single contig. So these are pretty impressive results, I would say, and compare very favorable to any other sequencing technology out there. So what can we see from these mags? How is this useful? So I wanted to draw attention to just one example here from patient 3 post-treatment data step. And in this example, what I'm showing is that we were able to identify definitively the probiotic that was present in this person had become engrafted as a result of the treatment 1 month later. So in this case, there were 2 hits to the biotherapeutic reference, which our industry partner has provided us with, and both of those hits landed on 1 contig. So together, there's a 2.75 megabase alignment with 1,625 mismatches, which is pretty great. And so all in all, what we have here is a single contig that is a 98% complete mag to one of the strains that they were most important in tracking. So that's just one patient. What about the rest of the patients in general? So what you can see here is that in all of these patients, we were able to detect, or almost all, we were able to detect the biotherapeutic number 1 in most of these post-treatment. And more than that, by focusing on the long genomic regions that were unique to the biotherapeutic strain, the HiFi data showed a lot of promise in being able to distinguish between the therapeutic and other closely related strains that might have been already resident in that person's gut. So what you see here is that almost all of the reads using this pipeline mapped to the biotherapeutic, as opposed to a different closely related strain that we were using to see if we could draw any of those off, right? Almost all of the data maps correctly to the biotherapeutic. And they are in the process, along with Richard and Dan, of developing a similar pipeline to assess whether the biotherapeutic 2 is present in the original strain, or if there are other closely related strains that are also present in the gut of these patients. Finally, there was no evidence for the long-term engraftment of biotherapeutic number 3 in any of the patients, but that was in line with results from previous assays. And it seems like for that particular strain, it dwells in the gut for only a short period of time, having an effect, but then not becoming fully engrafted. Another really exciting observation that we made as a result of this study was that, unlike NGS data, the vast majority of HiFi reads can be functionally annotated. So in this graph, we have all of the different patients that were tested, and then the different blue bars correspond to the annotations that could be seen using different databases and different tools. And then the dark blue bar over on the left is the combined results of all the possible annotation methods. So you can see that between 80 to 90% of HiFi reads have at least one annotation, and typically they will have 2 to 4. So what that means is that even though there were only 1 million reads for each sample, there were 7.29 million annotations on average for each one of these. In contrast, only 5% of the same sample NGS reads were able to be annotated, yielding about 2 million annotations per sample. So the HiFi data really outperformed NGS for this particular metric, right? And I think this is not really a big surprise to folks who are familiar with large genome assembly, because those folks know that annotation, the quality of the annotation is really limited by the quality of your assembly. Annotation is really an assembly problem. And for me, this really highlights the difference between data and information. So if the purpose of shotgun sequencing is to understand the functions present in a community, a complex community, then HiFi sequencing is a far more efficient method of getting that information. So what did we learn from this study? A number of things. First, that you can multiplex 4 samples in a single SMRT Cell 8M when looking at fecal microbiome samples, and you can get some high quality information from that, enough to be able to track strains, track engraftment, and to get some very excellent and deep functional annotation of these communities. You also see quite clearly that HiFi reads are really great at for metagenome assembly and binning, and producing a large number of high quality mags and even closed mags in every one of these samples. Second, you can see that there's very high resolution reference mapping, and so this is an appropriate method for following up on biotherapeutic studies. And then finally, you can see that functional annotation is highly efficient with HiFi reads. So I hope I've persuaded you that HiFi sequencing is a useful tool for metagenome studies. And if you'd like to learn more, I have some suggestions on where you could go. So first, you can visit the metagenomic page on the PacBio website, and there you can see all the detailed information you would need on library prep and also sequencing, and finally data analysis, including the pipelines that were discussed and presented here. And you can also see a wide array of publications from scientists just like yourself working on metagenome research, and also posters from various conferences. Another great place to go is the Shoreline Biome website. So on their website, they'll have all the information you need about 16S sequencing, strain ID sequencing and the different kits that you can purchase, as well as, again, a range of publications and posters that have been presented at recent meetings. Another great place to go is to check out the PacBio blog. So we've had a couple of really great posts recently. The first one here I'm showing is HiFi Advantage, Sequencing Advantage for Metagenome Assembly, and this presents the results of the data modeling, looking at the importance of very high accuracy for getting complete mags. And then you can also check out this great blog post that we have on 16S sequencing, which discusses the advantages of it, but also where you can go to find a PacBio service provider to do your 16S sequencing for you. Okay, so in closing, I just want to say, I wanted to point out something that I think we all kind of know, which is that the metagenome and microbiome research field is starting to mature. And I think one of the things that's coming out is that NGS sequencing alone may not be able to provide all of the information that we need to really generate the key insights that are going to be critical to making a difference for patients. And it's probably the case that we're going to need to look to additional sources of information in order to see more and to find those critical facts that we may have been missing that are going to allow us to make strong progress. So there's a number of different types of new information that are being explored. One is looking at metabolites directly, another is looking at proteins, and I hope I've persuaded you that HiFi data is another such really useful tool that you might want to look into in terms of moving your own metagenome research forward. Thanks so much for tuning in, and have a great day.